Dara, just let me know when we're going to go live.
Yeah, okay, so we're live right now and we are currently recording.
Okay.
We have 84 attendees. Maybe we give 2 more minutes.
Yes, let's please just start at 2 sharp.
Yeah.
Almost 100 people participating, but let's start as planned at 2 p.m. Central European Time.
Yeah, people are still alive.
Good morning, good afternoon, good for colleagues from different parts of the world that are joining this webinar. This is a webinar organized by the Global Antimicrobial Resistance and Use Surveillance System. And it's really a pleasure to have all of you attending this webinar today. That we will start in a minute. But just before we start this webinar, I would like to explain to you that this webinar is in the context of the revision of the WHO Global and Use Surveillance System, GLASS. As many of you may know, the global system was launched in 2015. And in these just about 5 years, we have right now more than 100 countries and territories participating in the global system. During this period of time, we were able to issue 3 annual reports, reporting on rates of AMR and also on the status of antimicrobial reference surveillance system. And the WHO has also issued a global report on the consumption of antimicrobials in countries from different parts of the world. So now, after this initial phase of the global surveillance system, it's true that we have achieved some success, but many lessons have been learned during this period. And it's really time to think about this early implementation phase and review the system to further develop and consolidate the global system towards better and more representative data, but also to explore the incorporation of new technologies that could enhance the antimicrobial resistance and consumption surveillance in countries and globally. So this webinar is today, it is a global webinar, and we would like to invite you to walk through the knowledge regarding the application of metagenomics for antimicrobial resistance surveillance. It is my pleasure to welcome Professor Frank Arestrup from the Technical University of Denmark, and Professor Arestrup is also the head of the collaborating center that is hosted in this university. So, Professor Aristrup, thank you very much for accepting to be presenting this webinar today, and I please give the floor now to you. Thank you. Just before I pass the word to you, I would like to also call attention that this webinar is being also recorded so that those who have missed it and would like to see it again, it will be available on our website. In addition, all participants are able to make your questions to Professor Aristrup through the Q&A window. So you should write your questions there. And we may also, if time permits, we may also have a few additional minutes for questions that you may also ask, always through the written questions in the Q&A window. So with that, without further ado, I would like to then invite Professor Aristrup to initiate this webinar. Thank you very much, Professor.
Thank you very much, Carmen, and thank you very much for inviting me to give this. And I also think that, as was just said, this is really also a continuation of all the activities that's been going on for what is it now, more than 5 years in the World Health Organization regarding how to improve the global surveillance of antimicrobial resistance, not only in humans, but also in One Health and in all other reservoirs. And we have been quite fortunate to be part of this journey because we have been at the WSO Collaborating Center in this area for the last 20 years. But today, the focus is how can we actually utilize metagenomics and analysis of urban sewage, and how could that potentially contribute to further improving the global surveillance of antimicrobial resistance. And before we start, we have, a slight, quiz. So there's a number of quizzes that have been embedded into this. So I would like to get your participation there as well. And this is the first one. And you'll have a maximum of, 1 minute to read this and answer it. But I think that, Daniel, you'll probably put the quiz up now, right? So 1 minute max.
Okay, coming up to 1 minute now. We've had 72% voted, which is great. 73%.
72% that has answered. That's good.
Up to 76. So I will end the poll now and show you the results.
Yeah.
There you go.
Okay, so it basically shows, I think, that's clearly see that quite a large number of the participants here are using both phenotypic and molecular testing to do AMR surveillance. So that is quite impressive. So good. But now we know, whom you are and where you come from. But there'll be more quizzes as we go along. Antimicrobial resistance, I think one of the things that we have to recognize is that antimicrobial resistance can have a very, very, very long evolutionary history. Traditionally, of course, we very much tend to look at resistance to last resort antimicrobial agents in critical ill patients at the hospitals. But resistance, that's not where resistance originally evolved and emerged. It could be in the animal reservoirs, wildlife, in livestock, in the normal healthy population that are carrying the infectious diseases before it eventually ends up into to human health problems and in critical care. So it really, antimicrobial resistance is one of these areas where we really need to capture the One Health approach and try to capture something, not only looking at the critical ill patients, but also at all the reservoirs and see how they fit together to get a better understanding of the involvement of resistance. So to be a little bit, slightly more, simply illustrated here, One Health and AMR, we perhaps have a tendency to look over here with the infected patients, but a lot of things are going on in terms of circulation in humans. And the thing is that in all these reservoirs, it might be different bacteria that are predominating, but the resistant genes that are moving around, they then will move around between different bacterial species before they eventually come out to become problems. And to really understand antimicrobial resistance, we do need to do surveillance of the genes and not only of the pathogenic bacteria. So, and it's also to address this, we also need to broaden our view in terms of where we do surveillance and for what we do surveillance. And the thing is, we have different methods to study antimicrobial resistance. And that can be to look at individual bacteria isolates, or we might actually want to go out and look at entire communities. Also, the bacterial communities of the bacteria, we might not be able to culture in the laboratories. We've looked at, just an overview of some of the different methodologies. I think the bacterial isolates, we're only looking at, a fraction of all the bacteria there, but we can do, plate counting, microbiota dilutions, e-test, disk diffusion, and, typically, with PCR or whole genome sequencing. But when it comes to the bacterial communities, which you might want to capture when we're looking into the reservoirs, well, of course, we can do plate counting, but that'll only get the cultural bacteria. And if you want to go in and actually find the entire diversity out there, we might have to go into PCR, metagenomic sequencing, or other kinds of activities. And one of the focus areas that we have is metagenomic sequencing, because it's really capturing everything. And then it also has the advantage that it will allow for reanalysis of the data if we identify novel genes at a later stage. I'm not so. The quiz number 2, to get a little bit more into details about what your institutions are doing. So which methodologies do your institution perform? And I think again we have these 30 seconds, 1 minute to answer the quiz. Go ahead.
Okay, I'll leave it for a few more seconds. We've had 180 people voted, which is...
Okay, that's good.
Okay, I'll end it and show it to you. There we go.
Okay, so well, I have to say that I'm quite, deeply, impressed that 29% actually do a whole genome sequencing, but otherwise, molecular test PCR. So certainly, at least within this group, the capacity to do some of these analyses should be out there and should actually be feasible. But I think that at least also the experience we have when it comes to some of the low and middle income countries that we work with, that capacity might not necessarily be there in all cases, and we really want to capture the entire world. But cool, it looks good. So what is metagenomics? And this is just for those of you that are not entirely familiar with the methodology. Well, it's a methodology where we actually go out and from a given sample, we purify all the DNA from all organisms, and then we just randomly get the sequence from small fragments. And then we can use that to identify and quantify what was in that sample. And we also refer that to, shotgun sequencing or random, sequencing. And this is simply where we take these random reads. And of course, you will get more reads from a bacterial isolate that is more prevalent compared to one that is not, and the same go for the gene. And shotgun refer to that this is random, just as when you shoot a shotgun against a target. And in this case, we really get the full capture, the full diversity of what is in any given sample, and can then afterwards identify what are the resistant genes there. It is not necessarily a completely simple process, but it's something that's actually capable. You can standardize it in any given laboratory around the world. But I'll just go, run through an overview of this methodology, the wet lab as well as the dry lab, because in comparison to classical phenotypic testing, then there's actually more on the bioinformatics side that needs to be done, and the computational expertise needed are more than when it comes to the phenotypic testing. But you normally start out with any kind of sample, wastewater or feces. Then you do DNA extraction. It has to be standardized for comparison sakes. You share the DNA into pieces. You do a library preparation, so you're ready. You put it on the sequencer, and then you'll actually have, the results. And the results will be all these small fragments of DNA. And millions and millions and millions of these fragments. But that has to be quality checked and trimmed. That has to be using standardized computer programs. It's something that is installed, but you could also use online services. We have for the last 10 years provided some of these, freely accessible online services that can be used for this. You can then use some programs to map to resistant genes databases and other databases to see how many resistant genes do I have in my sample and which are they. And then you'll get an Excel sheet or a spreadsheet out with that information from the farm or the feces, and then which kind of references. And then this is what we basically use in terms of making all the different plots and analysis of the data. And this is quite large amounts of data. So we would talk about several gigabytes, and you want to analyze everything. It very, very easily get into terabytes of data. So it can be sometimes a little bit disorganized, but there are standard programs out there that can help doing that. And there's also IT facilities that can help you in these kind of things. Metagenomics, we are very fond of it, but I also think that we should acknowledge that it has some limitations. There are certainly some benefits of doing metagenomics, but there are also some limitations. The benefits are clearly, from my point of view, that we go out and potentially get all the bacteria in a given sample and all the antimicrobial resistance genes there. So we're not missing anything, but we are actually sequencing everything. And we can, if we take, for example, from sewage, we can really capture something from very, very, very large populations with just one single sample. And very, very major advances is that once you have the data, you can go back and reanalyze it. So it's not like PCR that you have only identified, you only analyze for what you know is there, and then you have to go back to the sample to identify a new gene. In this case, you have the sequencing data, and they are very, very easily accessible, and you can reanalyze for novelly emerging genes. The limitation is that it has a low diagnostic sensitivity and specificity. So you're losing something on this area. There's not, as of yet, any good link between the given bacteria and the given resistant gene. So if you find a resistant gene in the sample, you don't know whether it's in a Salmonella, a Campylobacter, an Enterococcus, or something else. And it does require access to molecular techniques and high-performance computing. So that's certainly some limitations. And from a personal point of view, I would say that right now it is relevant for surveillance, especially of reservoirs, but I would also say that metagenomics still has limited relevance when it comes to the individual clinical diagnosis. Some people might challenge this, but at least with the current status of the technologies, I think that this is where we are at. So, quiz 3, and again, do you have these 30 seconds to answer? It's only 3 questions. So have everybody answered?
It's over 200 answered, 72%.
200 answers. Wow, that's really great.
Okay, I'll leave it for 10 more seconds and then. Okay.
Okay, I think that perhaps this was almost also a little bit of a trick question. So, but I can see that I can stop, with the sales talk right now. So, you seem to be convinced that this could actually supplement clinical surveillance. I'm happy to hear that, and of course that's a good point. But of course, we all have to recognize the limitations that are also of this methodology. But thank you. So, are you still? There it is. And so I'll just go to run through, an example. This is a pilot study we have initiated on this, something that we hope could perhaps potentially be an inspiration for how this could be rolled out under the control of the World Health Organization in the future. But as I said, this is a research project that is a pilot study, and of course not something that right now can be seen as the model for surveillance in the context of, for example, GLASS. But sewage has been something that we have looked at, and of course you could have said, why not soil? Why not feces from animals? Why not something else? But it's basically because it's close to humans, and then it's easy. It's easy to get permissions. Well, you don't need permissions. There's no sensitivity because the data are automatically anonymized. It's a way to monitor large, healthy human populations. And then it is basically, that the sample is already pooled, so we don't have to go out and spend the time pooling it. So it has all the advantages in the world. There can be something regarding contamination from different sources and animals and so on, but I really think, the advantages really outweighs what are the limitations of this. Sewage comes in very, very different forms, and this is pictures from some of the sites where we have done global sewage sampling, and it's anything from this site here, I think was in Nigeria, to this, I think was in Brazil. So it's, oh no, sorry, this one was Brazil. It's all different kinds of places it comes from. Initial pilot study results indicated that it was feasible to get a good coverage of the world, but also when we looked into the diversity there, we could really see that there was a separation of the world into the two different regions. That was a surprise for us, but of course we also have to trust the data. But at least for me, it told that this actually has some added value also in comparison to the clinical surveillance. It provides more information about what is circulating out there. We could also look into drivers for antimicrobial resistance, and just has been confirmed by several other studies. We also see that the Human Development Index and these social economic parameters is really very, very significant in driving the global occurrence of antimicrobial resistance. It is not only about antimicrobial use, it's also about these other factors. And this is really something that also needs to be taken into account in the future. And if we want to study the effects of intervening, then it will be very convenient to also measure what is happening in sewage in a given population. If, for example, one country, Ghana, invests heavily in health, well, does that then result in a decrease in resistance or something like that? We have continued this, so it was not only in 2016, but we actually have samples, and the last sampling round was here in 2020. So it's something we're continuing. We have up to 79 countries per time point, a total of 103 countries, 258 cities that has been covered over time. And we are also starting to make these draft reports, at least a suggestion for it, to see what is the developments in resistance over time. And it actually showed quite a high stability of resistance within a given country. And looking at the resistome, and this is getting very messy now, but we still see this separation in the different regions, so we can actually confirm that. But if we look into more details regarding this, not only total resistance, but let's say, what about if we only look at aminoglycosides? What if we only look at tetracycline? What was tetracycline down here? What if we only look at beta-lactams? Then what? And actually, so one of the things that we have been a bit surprised about is that we see that the world is separated in different ways depending on which resistant genes we look at. And I think this is certainly something that's, that requires further investigations in the future. We do not at this stage have any good explanation for this. We could see that the social economic drivers are driving this differently in the different regions, but it's certainly something that we want to capture and do something about resistance at a global scale in the future. We need to understand what is happening here. And this is also one of the advantages that metagenomic data have, that we can start doing these analyses. We also done a single within country study. And for us, well, in this case, we chose the US. So 44 sites in 8 states across the US, sampled in 2019 and 2020. And 20 was a little bit, complicated because of coronavirus, but it was certainly done at the same sites. But also here, we just very recent results, we actually see a little bit the same, that this correlation to the social economic parameters. So whether that is, people with health insurance, actually also average number of people per household is also significantly correlated to occurrence of resistance. So it at least confirms that at least within the US, these socioeconomic parameters are also somewhat, to a large extent, driving antimicrobial resistance. And this is a nice study that was recently published by Joachim Larsen's group in Göteborg, where they looked at the correlation between resistance in sewage and then clinical data. This is the 10 most common antimicrobial resistant genes in E. coli, and they find, as far as I know, positive associations everywhere. So it actually seems that there is a good correlation also between what you measure in sewage and what is actually a relevant clinical resistance. Still needs to be confirmed. I still think this needs to be done in a comparison for the future, but it's always, it's very, very confirming. It's very, very great that you actually can start seeing these, confirmatory studies. And so quiz number 4, and I think that perhaps there are, 2 questions here. So I think, 1 full minute might be appropriate here. So, but please go ahead. And remember, there are 2 questions.
Okay, I'll leave it for another 15 seconds or so.
Dara, I just suggest giving at least another 15 seconds because I have 2 questions here.
Yeah, sure.
Have you all answered the 2 questions?
Let's give another 15 seconds. We have 2.
Okay.
Yeah. We're up to 190 votes, so it's doing well. Okay, so I'll end it now.
Okay. So I think that's and that's actually quite a large. I think that's more than 40% would be capable of providing either samples or do the analysis. So I think that's quite positive. That 19%, and of course we cannot from here see what is the geographical or regional coverage of this, but I think that's actually quite alignment. Of course, need of training and equipment is high there. So I think point well taken. Naturally, this is also something that could require the involvement of national authorities, but it's good to know. And I can see that down there, if you could, then you could actually contribute, either once or several times a year. I think that's good, and it's also something that I know that these quizzes is something where the World Health Organization are trying to capture this information, because this is, I think as Carmen said yesterday, well, basically it's the member states, it's you that decides what, the World Health Organization should do. So I think that this is certainly something that's useful also for the GLASS program for the future. So thank you. And thank you for participating here. And so, but in case, if we want to go there and if we should go there, well, how do we actually get there? And I tried to make a few slides regarding what could be the next steps to do this. This will, of course, depend on whether people are willing to do this, and also depend on WHO leadership. And this is just my initial draft suggestion on this. So this still needs to be further developed. So please, this is a modest suggestion. This is not, the suggestion. This is just my take on this. I do think it's important to realize, and we also tried to highlight that in a publication early last year, that this is a complementary system to the current clinical surveillance. So it's not like, okay, now we can skip the classical clinical surveillance and we can make something new, and this has been stupid and we need to create something new. This is something for complementing the current one. It's something that could complement in the areas where the current clinical surveillance is, scarce. It's something that could supplement in terms of also getting larger healthy populations and not only clinical patients. So it is a complementary system that could hopefully work in synergy. I think that it's important we try to break down a little bit the process in terms of doing such a global surveillance. Well, it takes part, of course, you start out with, you have a sampling, DNA purification, sequencing, bioinformatic analysis, and then you need to do a report. And at least in my personal point of view, well, then global reports, that is at the end of the day, that is the responsibility of the World Health Organization. That has to be around that area that we have the responsibility. But how could it work? Well, I could of course say that, well, shouldn't we just make a local central facility that could do everything? It's a little bit the way that these pilot studies that we have conducted have been going on, where we have been collecting sewage samples from all over the world. Then we've done the DNA purification, sequencing, bioinformatics, and also provided the publications or the reports. But if this has to be something longer term sustainable, then that's, I wouldn't see that as a sustainable process. It has to be someone else controlling this. You could of course say that this is the way it has to be for a country with limited capacity, but of course the neat thing is that even if you can just provide a sample, just a sewage samples, then you could actually be part of the global surveillance. You don't have to set up a laboratory, you don't have to have a lot of expertise. Just if you can provide the sewage samples, you can actually be part of this. But if you already have some activities going on and you could do DNA purification locally, that would make some things a lot easier. We wouldn't have to do shipments of samples around the world. And then you could just take, okay, what about sending to a sequencing provider, getting the data, doing the bioinformatic analysis at the local site, and then you can start out writing the reports. You can also say that if you would prefer to do the sequencing locally or regionally, because we've also been taking part in setting up a regional sequencing centers in Africa right now through the Fleming Fund. So one of these regional centers could perhaps do some of the sequencing. Well, that could also be a possibility, and we could just share the data and do the bioinformatic analysis in a common way, a standardized ways, and then once again do the global reports. And of course, could also be that some countries would like to prefer to do everything themselves and just provide data into a common report, just as is done now for the GLASS reports. So, but I think that the advantages in thinking of this, that everybody can be involved immediately, and then as capacity develops and things are changing over time, you could actually implement something gradually more and more locally or regionally, or whatever might actually suit your system the best. So cost, and this is something I always hate, but it was something that was also asked by the World Health Organization to just put a little bit of a feeling on what would be the cost involved in this. And this is very broad estimates, so do not necessarily take this off at face value. This also excludes any kind of, central collating of the report or writing of a reports. So there's of course something that's not including in this. But if you want to do everything, well, you need to have the capacity to do the sampling, the shipment, purification, sequencing, bioinformatic analysis, and of course that will come with a cost per sample, and as well as a cost if we say 500 samples from 200 sites in 100 countries. That will be a reasonably global surveillance. Well, the expertise for sampling and shipment is very, very limited. But if you want to do DNA purification, you do need to have a microbial laboratory where you can do this in. And there is the need for a certain amount of equipment. If you want to do sequencer, well, you need to have the capacity to do this, the human capacity, as well as access to a sequencer. And a sequencer can be expensive. Bioinformatics, well, for metagenomics, there is no way around that you do need a computer above the size of an average. So this is normally what you call a high performance computing. In our case, we actually, we personally use, well, we do not do sequencing inside. At my laboratory, we actually ship it off either to China or the US because it's much cheaper. We couldn't dream of doing the sequencing ourselves because there are sequencing providers that does this much cheaper than can be done in-house. For high performance computing, we also use a central facility. So I'm not necessarily in favor of buying computers and sequencers everywhere because it's actually quite expensive and it is not really necessary. As I said, we do not use this at our university ourselves either. But if you want to do everything locally, then there's some investment in equipment. For DNA purification, this is very, very minor. And of course, there'll be some expensive per sample. But a sequencer, to do this, well, you probably need, a $200,000 investment to be able to just do two samples a year. That would not be cost effective. It will be expensive, but and this is the neighborhood of what we'll be talking of. And if you want to also do the computational cost, depends a little bit on what you need, but it can range very, very easily. But a minimum of $200,000 would actually be the investment needs to do this. And so the cost per sample in terms of equipment would be staggering if you had to do everything at hundreds in the different laboratories around the world. And just to calculate it up, it would be, an investment of $40 million US if everybody needs to have the same capacity. And I think that might not be feasible if this is the way we want to go. Another option is, of course, to run something where everything is centrally. This is actually a mistake. This should not have been here, so please ignore these 5,000 right here. And this is just, if you have a center facility that can do everything, then this should be the cost. And a total cost of generating the data would be less than $200,000 on an annual scale. But it could also be, as I said, other ways of doing this. So it's not everything that's central, because there will also be countries that will be reluctant for practical as well as administrative issues to send biological samples around the world. I think there are good reasons to do this. I know right now that there are some countries that are very reluctant to send sewage samples because they are concerned about if other countries will start looking into a SARS-CoV for this. But if you do the DNA purification, which would exclude any kind of RNA work centrally, well, then you would avoid these kind of problems and you would still keep the control. And so one way could be to do at least some of the handling locally, and then simply just, as we are, use a sequencing provider and then get all the data in centrally into some kind of HPC facility. I do think that if it should run, it should be something that's controlled by the World Health Organization, and it would actually be quite cheap in terms of cost per sample, much cheaper than doing it centrally. There will be some investment cost, but I think that's seeing that the answers you just provided, I think that most of you would already have this capacity. So this would be much less. I think it will more than 50% of the laboratories that could do this. So you can divide this by 2. So it would be an initial investment, but then actually a relatively limited amount of funding that will be needed to run this for the future. But of course, this is very broad estimates, and just to give you a feeling that there are different options and they come with different costs. And so step-based building, but contributor for day one is something we would like to suggest. And you could, if you have no capacity, but you could just deliver a sample, you can deliver DNA, you can start delivering data, and you can analyze the data. I do think that one of the big advantages is that everybody could contribute, but also be giving access to analyzing the data. And I think that that will be a benefit for the entire world and the global surveillance of antimicrobial resistance. So thank you. This is the presentation from my side. I'd be happy to answer any questions. I'm not sure I can answer everything, but I'd be happy to try. Thank you.
Thank you very much, Professor Aristrup, for this brilliant presentation. I really invite and motivate participants to add questions either to the Q&A, or if you have any difficulty, to add your question to the chat. But while we wait for specific questions on metagenomics for Professor Aristrup, I think there is a very interesting question in the chat that I prefer to respond live and to all of you. So the question is it worth to continue surveillance for AMR while the pipeline of effective therapies is decreasing significantly? We have been screening for AMR for decades, since the 40s, while bacteria will never obey us and be sensitive. In other words, how beneficial is the surveillance efforts in tackling the AMR challenge? This is an interesting and provocative question, and I think that we have to consider that it was exactly when the policymakers, politicians and the policymakers in the field of health and other fields, it was exactly when we could provide some surveillance data on the potential impact of antimicrobial resistance, not only on the health of human beings, but health of animals, especially for food animals, the health of the environment, and the impact it would have in terms of development of humanity, to the point that the current sustainable developmental goals, they do include in the agenda the monitoring of antimicrobial resistance as an indicator on whether we are being able to tackle this risk to humanity. Plus, it was really because of some surveillance data that we could call the attention of countries, member states, governments, to then invest more in the production, exactly as the colleague has raised it, to invest in the production of new therapeutics. And the WHO issued a list of priority pathogens for research and development, exactly to address the gap due to the dry pipeline, as we see. But I think that I take the point of our colleague that it's not any type of surveillance that we need. We need to know much more. We need to be a bit ahead of the bugs. So number one, we need better information on who is being affected and how badly the individuals or animals are being affected, because then we can mitigate at least the impact of AMR in these populations. But also we need to be ahead of the game in terms of detecting resistance mechanisms that could be informing in advance the research and development of new diagnostics, but also new therapeutic options. And in this regard specifically, I think that the methods that give access to the full genome of the pathogens will be really an advantage in putting us a bit ahead of the curve and being able to better inform research and development. But I see, Professor Aristrup, that while I was talking here, several questions came through this, the Q&A. Actually, we have 10 questions now, and perhaps I would like to let you go through the ones you'd like to start answering, and I will also be screening the questions. Over to you, Professor.
Yeah, so I think that thank you, and I do appreciate that the first question was, perhaps a little bit provocative, but I think it's very nice to get, these questions that are challenging our current view of saying surveillance is just good. But I think, as Carmen said, it has certainly been useful and used to a large extent to also do real-life intervention, and has been used to reduce antimicrobial use as well as targeted intervention against antimicrobial resistance in many countries, especially Europe and North America. And I also think that without surveillance data, we would not today have recognized that it's not only antimicrobial use, but it's also other factors that contribute to the global occurrence of resistance. And I think this should be targeted for intervention for the future. But I think that there was one question regarding this about the central facility. It has just disappeared, but there was one that asked whether there is a central facility that could handle samples from all different reservoirs and then do that. Certainly, that is feasible, but I think that this needs to be organized in a good way, where there's someone controlling what it's doing and also controlling the flow of the data. So it's not just that the usual thing that I sit here in Denmark and you send the samples to me and I'll just utilize it for whatever I want. That's not a good way. There needs to be some kind of governance around this, and I think that governance has to be it's national public health, and that is the World Health Organization. So because I wouldn't like to misuse your trust, and you wouldn't, of course, like to misuse me. But then there's also a cost around this, and there needs to be someone that's actually addressing that. And then regarding the next question, could the World Health Organization support implementation of metagenomics technology in my country, technically and financially? I think that, of course, the financial part would be for Carmen to answer, but technically, I think that we can provide guidelines, yes. But if you looked at the numbers I calculated, then if you want to do everything inside your country, it would be impossible for the World Health Organization to fund that. It's a very large amount of resources, and I think that perhaps we should be smarter in utilizing this. And it doesn't really make sense to set up sequencing for metagenomics in all countries in the world, and depending on what is actually the use of this. I think we should utilize the resources in a much smarter way. And then there's another question. If we have the local capacity to sequencing and bioinformatics, how could we articulate how will the interaction among countries? I personally, but again, I give the floor a little bit back to the World Health Organization later on. I think that the good thing could be if we follow the same standards and protocols, and then we need to feed these data into a common database where we actually do the same bioinformatic analysis there. I think that's actually key. We need to do that in parallel. And then I think that we could then utilize this just as data, just as if it was generated data in a single laboratory only. I think that is really the perspective there. And then from Michael from Canada, is there any plans to examine antimicrobial residues in wastewater at a global scale? I personally do not have any plans to do that. I know that there are people that would like to look into this. I think that it's interesting. It's certainly also relevant, but of course, we also know that we're going to miss something like the beta lactams because they're degraded in the environment. And this is, after all, 50% of all the antimicrobial agents used. So I think that, yes, it could be done, but I'm not aware of any plans. And then another question, is it possible to get AMR data in wastewater in individual country to understand the individual country situation? Yes, I think that is what we should do. Exactly, we should start getting, basically as many wastewater and crop resistance data from as many countries as possible, and get that consistently over time from these countries to see, is the resistance situation stable? Is it going up? Is it going down? I do think that when it comes to antimicrobial resistance, then it's decrease or emergence that is not going to happen during weeks. This is actually something that happens over very, very long time periods, especially if we want to see it's going down. So I think that is something that I'm encouraged that we should start doing. And yes, can I elaborate more on the plans to build capacity for institutes, countries with NGS capacity, but limited metagenomic analytic capacity? I think that from our sides, what we try to contribute with is knowledge capacity. We do not have any funding whatsoever to say, okay, now we can buy equipment and so on. That has to come from someone else, or it has to be on your national funding. But we have developed a number of online courses in this, and we are right now, expanding on that and making another one. So one of the things that we are focusing on is to try to make online courses that then can be assessed by everybody, and hopefully that can contribute to this. And then, thank you for a great presentation. Have you correlated the phenotypic data from sewage with the genotypic data? Have you looked at the flow across different countries? We're looking at the global surveillance. Well, I think that if you look at this correlation, then there was this very recent study by Joachim from Sweden, where they had looked at the correlation, and that was very promising. They just published that. We have also done it. We compared to other phenotypic data, and we see the same, that there is basically a good correlation between what you find in sewage and what you find in phenotypic data. We also have just done a global collection of clinical isolates in 45 countries. Some of you might have contributed to that, and I'm sorry, but I didn't have time to get into this. But this is also where we would like to, in these 45 countries, 90 hospitals, we are getting the NGS data on single clinical isolates, and this is also looking how good is the correlation actually to the sewage data, and hopefully this will further support what we've seen so far. And looking at the global transmission, yes, but looking at the flow is of course impossible when you just do a single point prevalence. You can look at similarities in terms of genes, and we are also doing these more advanced analyses where we're not only looking at the genes, but also what is the context they're sitting in, so which bacterial flanks, what's the flanking regions. So we're doing these analyses, and hopefully that will tell us more about the global dissemination and the dispersal limitations of AMR. But I unfortunately did not have time to get into this during this talk. Is AMR metagenomics enough to characterize the genes, or the taxonomy classification is also an important step? For the AMR genes, then the resistome, that's sufficient to get the specific resistant genes. We can get that relatively precise, but there is a limitations in terms of we cannot tell whether that resistant gene is within a given bacterial species. At least that's more difficult, especially if they are on plasmids. Then we might be able to tell they are on plasmids. So there are some limitations, but I also think this is why this is something that is supplementary to the clinical surveillance. It's not standalone, it has to be a supplement. In the context of One Health, would sewage samples be informative of environmental AMR? My guess is absolutely yes, and I do think it would be a very good sampling frame, because you could also say that you go out and sample soil, but if you sample soil where you have added sewage or you have added waste, just in the last few days compared to looking at it 1 year later, you're going to get different results. Whereas sewage is easier to standardize, and this is at least what is being disseminated out into the environment. So this is the way we and our waste is polluting the environment. So I do think it's a good sampling frame also for the context of the World Health aspects. In the ideal world, we would combine this with samples from animals, at least the main livestock species, but there is of course also a limit to how much funding is available out there, especially if you have to do constant surveillance over time. And then a question, very interesting talk. I'm originally from Afghanistan. I'm wondering if you have any plans for this country and this year, how we can help. Well, my hope is that all countries in all, 203 countries in the world or whatever we have, would actually be interesting in participating in this, and they would be happy to provide samples, and we would have the funding and energy, you know, to analyze it and share the data with everybody. So I think that it will be naturally very, very, very great if Afghanistan, you know, is one of those countries, because it's a big country, but certainly also a country with some special issues, socio-economic, that could be very interesting to look further into. And I think that so having a within-country sampling of that country of Afghanistan could be very intriguing, just as we did for the US. And then thank you for a very nice talk. Do you have any examples where metagenomic data led to specific interventions? Well, trying to think of it. Besides the Denmark, that's the swine population, where we have started to make novel restrictions on how you know, the consumption of antimicrobial agents are based on metagenomic surveillance of the swine population. Then I think that's it. I do know that partly based on our first pilot data, then the World Bank in their report suggested that perhaps the focus should move away from just antimicrobial uses to also these socioeconomic parameters. So I think that's certainly also part of the impact. But metagenomic surveillance is very novel, so of course it hasn't had the time to get to reach its full impact. And then should metagenomic surveillance be implemented rather periodically to answer the surveillance questions which can differ over time? I'm not quite sure I understand the questions, but I think that what you need to do with surveillance is that you need to implement it and then do it consistently over time. If you wait until you have done an intervention or something is happening, then you're too late, because you need the a priori baseline. And I think that that's always a challenge. So the sooner the better. So I really think it'd be great if we could start implementing more surveillance across the world, and then, you know, capture these natural interventions that will hopefully happen, because that's where the true value of surveillance will actually show itself. Another question. While metagenomics enables you to detect all DNA in a sample, how can we further link the genes detected to the risk assessment framework, or if it is possible? It's not better to focus on a uniform culture standards, and of course the lack of standardization. Depth and data processing is difficult to compare international. You have a very good point there, and to a certain extent. And I'm a great fan of a very, you know, a good risk assessment frameworks and so on. But I think that we also had a recent study in the food control. We're starting to actually address that if you can, by metagenomics, tell something about what is the occurrence of resistant genes in different reservoirs, then you can at least use this to start making, you know, exposure assessments. And you might eventually also be able to use this for source attribution assessments. So yes, it's complicated, but it's not undoable. And I do think that these things, you know, could go well, extremely well, hand in hand. And then thank you to you know, that's from Mohammed, you know, to Carmen for the answer. Yes, and good, and once again a good question, I think. And what do you think are the main drivers behind the relation with human development index variables? Is you again, or more related to spreads? This is regarding what is the social economic parameters that are driving the recurrence resistance together with AMU. Our best guess right now is that this is basically, if you have circumstances that are doing that, you have a very rapid transmission, or if you have poor hygiene, you might say, and so you have transmission of anything, then antimicrobial usage is still the Darwinian selection for resistance. But then anything that will increase transmission will make sure that this selected resistance will spread and can be further selected. In other words, 1 kilogram of antimicrobial agent in one country might, you know, add very little to resistance if you have a very good hygiene, where in other countries it might, you know, actually it really adds to the antimicrobial resistance. So for me, that is the most likely reason behind this. But this is something that we and others are looking further into now, amongst others also using these global sewage data. And then, do you or will you test the same samples or aliquots for residues as well as resistant genes? We do not have any plans to do any additional testing for antimicrobial residues with the samples we have right now. We did that together with a partner in the Netherlands for the first pilot studies, but we have not had any plans to do that right now. Oh, hi, Jaap. So how standardized is currently the output of DNA isolation and sequencing? Or is the risk of clustering data from different locations for technical reasons? Yes, absolutely. I do think that I also mentioned that a number of times that standardization is really, really, really important here. I do think that it is as a standardization using the same equipment, doing ring trials. I love ring trials, and ensuring that the quality is good. That would actually lower this. But of course, if you do local DNA purification, then you're going to get some kind of a local variation compared to it if you did everything centrally. But there's also a variation that will happen if you ship the samples from different countries. So I think that at the end of the day, the decentralization of the DNA purification is probably okay. If our country, Algeria, has decided to join this project, how would it help? As we could extract DNA, and we have sequencing in the health institutions also. Cool. I think that'll be great. And I think that this is also something I really think that I point a little bit now to WSO, Klaas, and Carmen here, because if this is something that should run from the outside, we have this small pilot project, so we run it at least for the next year, what we are already doing. But if this has to run into the future, we need another way of collaboration, and there'll be a governance. We need someone to decide who's going to be allowed to do what and when, and I think that should be the World Health Organization. So, then a question: how do sewage data correlate with the food safety threat of veterinary drug residues as observed in agricultural products such as meat and eggs? And when I think sewage data is the greater contributor for AMR spreads than directly coming from food or animal origin. The first question, I think that is two different questions. I don't think that we can tell anything about the veterinary drug residues in human urban sewage, but perhaps if we looked at slaughterhouse sewage, that could be a possibility, or samples from slaughterhouses. And the contributor, whether it is sewage or spread directly from food events, I think that very much depend on the country. And I think this is also along the One Health concepts where you do the surveillance country basis and then try to figure out what are the most important drivers. And then, is there any correlation or study on metagenomic data and the role of food animals as a reservoir for AMR? I think that the best study we had, that was actually something that was coordinated by Jaap Wagener, that I can see is also on the call here, and looking at metagenomic data from food animals all across Europe. That was an exposure assessment study, you might say, saying, okay, what could come from where? And there was also embedded in that a number of detailed longitudinal studies from the Netherlands on, transmission of resistance into the human gut. I think what was lacking there is that there are more long-term studies where you look at what is happening in a country during 1 year is probably impacting clinical resistance the next year. And I think that we still lack these studies. I think that's, of course, again, call for more continuous long-term surveillance so we can start getting these data. And then sewage surveillance is the rule of the health ministry or the environmental minister. That depends on the country. I think that in my country, Denmark, then sewage is considered an environmental sample. And basically in Denmark, you can do anything you want with an environmental sample. There's no restrictions, there's no permissions, you don't need anything. I know that in Tanzania, in order just to do any analysis of also environmental samples, you need a local ethical permission. So I think that very much depends on the individual country. So how is this the initial difference or complement to the global antimicrobial surveillance system? I think that's it's a complement. It's not really different. It's a complementary to it, because GLASS is clinically resistant, and what is available on a clinical resistance from the countries. And then if you could supplement that with a more environmental or population-based surveillance based on metagenomics, I think that would go extremely well hand in hand. And may I suggest publishing your full protocol so people can follow it? That has been published. So that our protocols should be available. If there's details that are not available, then I'm sorry. We are working on a full protocol that goes a little bit more into details, also how we do the bioinformatic analyzer will do that, but the initial step is actually already public available. And you can go into this homepage. I showed on my first slides, and I think that you should be able to find it. A more full protocol with more details will follow shortly. How possible is it to start and promote field testing and sequencing for AMR for sewage samples? I think that, well, perhaps all of you are in a much better position of answering this than I am, because I know the difficulties for my country. You're the one that know the difficulties are potential for your country. I think it's be feasible, but again, I do think that we need a good governance around this. And once again, pointing to the WHO, because they are the global governance on this. And can you share a link from the first slide again? Yes, I'll try to do that. Do you think, proximity ligation 3C is as effective as metagenomics software for monitoring and surveillance of AMR? I have absolutely no idea what is proximity ligation 3C. I might have, but you might be using a different word for this. But there are, of course, a number of different ways of you could go out and analyze these data. You can use also qPCR. There's also resistance cap. There's capture sequencing approaches you can do. We have preferred metagenomic sequencing, and it's also because we think that this is something where we can reanalyze the data for novel resistant genes later on. But of course, that is a matter of choice. So, but I think that's, there's another question, I think that from Joachim. Hi. COVID-19 surveillance has become very popular. Yes. I see any challenge interpreting such surveillance data before, rigorous comparisons with samples from individuals in corresponding populations has been made. Yes, I do. I do think that we actually also ourselves doing that a bit, and we are right now analyzing 1,000 sewage samples from various countries for COVID-19. COVID-19 or coronavirus, SARS coronavirus 2 is much more susceptible to the physical handling here. So it's not as if this was enterovirus or something that would be easier to standardize. So I think the standardization around handling this. I would even be a little bit reluctant to compare data from different laboratories there, especially in terms of the qPCR, so the amount of data you get. Whereas I think that the sequencing data could probably provide pretty good results there. Erasmus Medical Center had a very recent publication that's just been accepted in emerging infectious diseases on this. And I think that might be worth looking into that. I'm a bit biased there because I'm one of the co-authors, but I do think that it's probably more relevant for determining something about the genomic variation in the circulating strains than as per se a quantitative measure. But I do think it is very much like a case-by-case thing you have to look into. And then having access to metagenomic data will also include access to human genomic data. What are you doing to assure the privacy of data? And I can think that's very easy to answer that. Absolutely nothing. Because that was one of the first questions we addressed. And I know that ethical considerations might differ from country to country, but the big advantage of sewage data, that is that it's automatically anonymized. And in that case, you cannot trace any read or any sequence back to an individual. And at least according to the way you consider ethical concerns in most countries I'm aware of, then there are no ethical concerns there. You do not need any ethical approval or anything. You can basically do anything you want with these data. And it's one of the first questions I asked our National Ethical Committee in Denmark, and there was a very clear answer on that one. We have even looked at human genomic data in these samples, and that should be without any problems. And then, yes, I think I answered the last, I tried to answer the last question, but what is, Mamitina, please send me an email if that was not sufficiently good an answer. So I think that I know that was fast. I hope that I didn't lose anyone here, but yeah.
Thank you very much, Professor, for these very extensive answers that you have provided to all questions. I think that you parked some of the questions and actually you forwarded them to me somehow. I am conscious that we are already beyond the time, so almost 10 minutes past, but I would like to highlight two things in regards to some of the questions made by the participants. The first is about the support the WHO can provide to countries in regards, for instance, to metagenomics. Well, I think that the very first step for WHO to provide support is the country decision on what the country would like to implement in terms of the surveillance to combat AMR. And note that I'm talking about in terms of surveillance, that is not only surveillance of antimicrobial resistance, but also surveillance of antimicrobial resistance drivers, such as the use, consumption, and other possible drivers, like the ones you have, Professor Frank highlighted, the lack of infection prevention control strategies, the lack of water and sanitation that can promote the emergence of antimicrobial resistance. So I think that the first step is the decision of the country. And in fact, it is the WHO role to be working with countries to support the development of capacities and the development of surveillance systems to monitor AMR and its drivers. And in fact, the Global Antimicrobial Resistance and Use Surveillance System, GLASS, the main objective is to foster the development of national surveillance systems in countries. And as a matter of fact, out of the more than 100 countries nowadays participating in GLASS, more than 1/3 of these countries did not have any surveillance system in place. And the starting point has been indeed with clinical samples and using phenotypic methods to evaluate the presence of surveillance, of resistance. But indeed, AMR is very complex, and we need several approaches to understand, to better understand this problem. And I think Professor Ari's group has very nicely responded that there is no contradiction between the metagenomics that he presented, method, and the global surveillance system. We are just here talking about new technologies and new approaches that could complement the surveillance efforts. Now, in terms of incorporation of new technologies or new methods in GLASS, I have to say that GLASS is really driven by our member states. And that's why right now I started this webinar saying that we are in the middle of the revision of the global system, and the revision is done with the representatives from countries. So the next steps of the global surveillance system, including the incorporation of new technologies, such as the new sequencing technologies, will be also discussed and decided with the countries participating in GLASS. So we do hope that in the near future, we'll be able to then come up with some suggestions, some framework for incorporating these new technologies. So I am very mindful of the time that we have really exceeded our planned time, but it just shows that the success of this webinar, and I'd like to really thank very much Professor Aristrup for this brilliant webinar that has motivated so many questions and debate. And this debate should continue. I think that today, with this webinar, is just to give you the message that GLASS is indeed looking and exploring how to incorporate technologies such as the metagenomics and what could be the approach, etc., in the framework of the global surveillance system. We will certainly need to come back to you, and we need to be working with Professor Aristrupa's WHO Collaborating Center, as well as other WHO Collaborating Centers, and many of you in the audience today for continue to walk this journey with us. So really, thank you very much for your participation. And Professor Aristrup, really, again, thank you for your very generous contribution to this discussion. Thank you.
Thank you.
With that, I would like to say goodbye to all of you and thank all of you for having joined this webinar. Thank you.